Welcome to this biochemistry lecture on gluconeogenesis, the Cori cycle, and ketone bodies. Kind of a hodgepodge of topics that are related to glycolysis, and so we'll try to cover them in one sitting. So as we've noted, we're going to talk around processes that are around glycolysis. Gluconeogenesis is almost a reverse pathway, and so that's what we're going to talk about, and they're all around the glycolysis process, just to give you a big picture idea. What happens is gluconeogenesis it generates glucose from lactate and other carbon substrates like glycerol and amino acids, glucogenic amino acids. And this happens when your muscles start producing lactate and alanine and have protein degradation. This process that happens in the liver will restore glucose that can go back to your muscles. And again, it occurs mainly in the liver and a limited extent in the kidney and small intestines. And synthesis of glucose from pyruvate is it's fairly similar to the reverse reaction of glycolysis. Only 3 reactions have a large delta G, so they are essentially irreversible. So that's hexokinase, phosphofructokinase, and pyruvate kinase, and these steps we kind of bypass, and they have their own enzymes, which we'll talk about. I can talk about it right now. So on the left, you have glycolysis. On the right, you have gluconeogenesis. And if you go down step by step, you can see exactly the enzymes that occur. And a lot of them are very similar on both pathways. They're just reversible. So if you pick, triose phosphate isomerase, it's the same for both. It's just which way the reaction is going, which way the pathway is going. There are a couple, as you can see, like fructose 1,6 bisphosphate, whose sister enzyme is phosphofructokinase 1, that you can't do that with. Most of them are, again, kinases. Hexokinase is the first step of glucose or glycolysis. Glucose 6-phosphatase is the last step of gluconeogenesis that gets it to glucose. So these are the main steps and the differences. I would know these enzymes and where the differences between the two processes are. This is, to prevent counterbalancing each other, glycolysis and gluconeogenesis are reciprocally regulated, meaning if one shut off, the other one, or if the other one's going, the other one's shut off. Okay, so there's several control mechanisms. Usually it's by adenine nucleotides, and that's the mechanism by which they're regulated. So there's a couple of them. There's cyclic AMP cascade. That's triggered by the glucagon. When blood glucose is low, that's a trigger to switch the process. Phosphorylation of enzymes and regulatory proteins in the liver also can result in inhibition of glycolysis, and that can stimulate gluconeogenesis. So some of these enzymes get phosphorylated, and the reverse reaction occurs. Some of the regulated pathway or enzymes are protein kinase that are phosphorylated by protein kinase A, which is pKa usually. So pyruvate kinase is inhibited when it's phosphorylated. CREB, which is cyclic AMP response element binding protein, is activated to transcription for PEP carboxykinase, and that increases gluconeogenesis. You have bifunctional enzymes that make and degrade allosteric regulators, so fructose 2,6 bisphosphate is involved. Anyway, so any number of enzymes can be involved in regulation. An example here is phosphofructokinase is inhibited by ATP and stimulated by AMP. Fructose 1,6 bisphosphate, which is the reciprocal enzyme in gluconeogenesis, is inhibited by AMP. So they both are regulated. One's inhibited by AMP, the fructose 1,6 bisphosphate, where the other one is stimulated by AMP, the phosphofructokinase. So that's pretty much, they have the opposite effects with adenine nucleotides. Again, and that ensures that when ATP is high, right, your glucose is not degraded to make ATP, and when ATP is so that, and then it goes more of the glucose will go to glycagon, and when ATP is low, the cell does not expand energy in synthesizing glucose, which is the gluconeogenesis pathway. So that's kind of how everything's kind of self-regulated in this environment. Phosphofructokinase is the rate-limiting step of glycolysis. It's allosterically inhibited by ATP. That's a very important concept for you to remember. A high concentration of ATP binds at a low affinity regulatory site, and it promotes the tense conformation, which has significantly less PFK activity. So that's an important note, and you can see that by the activity curve that you have. So when ATP is low, you have the blue curve. When ATP is high, you have much more. You have a significant lower rate with phosphofructokinase. If we have an allosteric regulator, fructose 2,6 bisphosphate also has a similar effect to low ATP, and you can see that the relaxed state is activated again in that situation when the ATP is high. So the PFK is allosterically regulated by fructose 6 phosphate, and you can see that this shows a ribbon structure or a cartoon structure of the PFK, and it binds to the fructose 6 phosphate. So, and that allosterically regulates that reaction. Cyclic AMP dependent phosphorylation is important also in the phosphofructokinase. So you have, if it gets enzymatically activated or phosphorylated, I'm sorry, then if it activates the FPBase2, it also inhibits the PFK2 protein. So fructose 2,6 bisphosphate decreases in liver cells and in response to cyclic AMP signaling, and is activated by glucagon when the blood glucose is low. So all these downstream effects include glycolysis slows down because of too much fructose 2,6 bisphosphate is not available to activate phosphofructokinase, and then gluconeogenesis increase because a decrease in concentration of that fructose 2,6 bisphosphate inhibits the gluconeogenesis enzyme fructose 1,6 bisphosphate. So there's regulation on both sides depending on what's going on. And it's reciprocally regulated. So again, this kind of gives a summary slide of how the cyclic AMP cascade in the liver works. So if the gluconeogenesis is stimulated, glycolysis is inhibited, and that's all has to do with how the response to the general signals from cyclic AMP. So cyclic AMP cascade is initiated, you get glycolysis, gluconeogenesis is stimulated, and glycolysis is inhibited. And you can see how this is regulated in that pathway. And there's I'm just throwing this in there for the sake of an interesting note. You'll note that the glucagon receptor and the adrenaline receptor are GPCRs. And as we talked about before, those are the major drug targets. They're also the major receptors. And so they play an important part in every part of these processes. And I'm willing to bet that there's things that we have I know that we have things we have no idea what they are. So now I'm going to talk about the Cori cycle. And I've already described the Cori cycle to you. It's when we have pyruvate and glucose get burned into lactate. And that happens under anaerobic conditions. We didn't name it the Cori cycle, so I'm going to put that in there as just to label it so that if you see it on a test or somewhere you know what that is. Generally speaking, that we burn, it happens when anaerobic conditions occur when you're exercising. It actually costs to burn energy this way. And so it costs 6 phosphate in the liver for every 2 that are made available in the muscle from that cycle. So the net cost is 4 bonds. And although it's costly, if you're under anaerobic conditions or in need of skeletal muscle, we spend it. Now the other thing that can happen, there's 2 other things that can happen when we have this. So in between, when we start going from glycolysis and going into lactic acid, we can use phosphocreatine as our phosphate. And we can also have this issue during marathon running. And so that you'll see that situation occur when we get to that situation. Cancer, we use this cycle when the tumor development is faster than blood vessel, and so blood vessels will shift to this too. Our cells will shift to this to keep up with their energy demand. Ketone bodies, that's another source of energy. So acetone, beta hydroxybutyrate, and acetoacetate, they're formed primarily in the liver mitochondria. They can be used as fuel in most tissues and organs, and the formation occurs when we have an excessive acetyl-CoA in relation to the amount of oxaloacetate available. So this happens when your diet is mainly fat and low in carbs, or when your diabetes is in control or starvation type conditions. So in fasting or diabetes, gluconeogenesis is activated and oxaloacetate is consumed in this pathway, and you'll get fatty acids are oxidized, producing a lot of acetyl-CoA, which is converted into ketone bodies. That ketone bodies then are metabolized into beta hydroxybutyrate and acetoacetate and acetone, and to give the energy to the cell that is necessary. And they diffuse from the liver mitochondria into the blood. They're transported in the blood, and then they're used as energy. Heart muscle and renal cortex use acetoacetate. They prefer it over glucose in physiological conditions. The brain adapts acetoacetate during starvation and diabetes. So these are not uncommon molecules to be digested. So oxaloacetate is normally formed from pyruvate by pyruvate carboxylase. Fats burn in that's the cause of it. Again, the entry of acetyl-CoA into the citric acid cycle depends on the availability of this oxaloacetate. And so the concentration of oxaloacetate is lowered if it's not available, if carbohydrates are not available, like as you'll see in starvation or in diabetes. This is why you get diabetic ketoacidosis, where you get ketone bodies that form as a byproduct of this process, and that process then increases the acidity in your blood and causes other problems. 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA. That's a thiolase reaction. So the last one is if we have synthesis of 3-hydroxy 3-methyl-glutaryl-CoA. If acetyl-CoA reacts with water and acetyl-CoA to give us 3-hydroxy 3-methyl-glutaryl-CoA and CoA. And the enzyme is HMG-CoA synthase. And from there, we get acetoacetate. So 3-hydroxy 3-methyl-glutaryl-CoA is cleaved to acetyl-CoA and acetoacetate. Those are the reactions that occur. That's done by enzyme HMG-CoA lyase. 3-hydroxybutyrate is formed by the reaction of acetoacetate by hydroxybutyrate dehydrogenase. That's a pretty straightforward reaction. You probably just need to memorize it. Acetoacetate also undergoes a slow, spontaneous decarboxylation to acetone. So you can tell if someone's in this condition because you'll smell acetone, which is fingernail polish remover. You can detect it on the breath. This is how dogs can tell if there's a diabetic shock in people. Actually, diabetic ketoacidosis. That's the lecture. Look forward to hearing any questions. Thank you for your attention.